Thank you. So I chose hepatitis delta because it is something not people cover, right? Because it's usually a rare disease, most people may not see delta. And also, I wanted to highlight some of the work done by one of my colleagues, Arshi Khanam, who is a alumnus of ILBS. So hepatitis delta is an incomplete virus, the smallest virus known to man that infects human beings. It masquerades itself with hepatitis B. So the surface of the hepatitis delta is always of hepatitis B, the surface antigen, but the nuclear capsid contains the delta antigen. That comes from the delta. So it just hijacks the hepatitis B envelope and then carries around as a hepatitis B delta. It's a very unique mechanism, and that's something that the only virus that infects human beings do that. So it was identified by Mario Rossetto, and in very long time ago, 1977, he was suspicious of some of these people with acute hepatitis may have something else other than hepatitis B. So we looked at so what he did was he actually figured out a way of looking at cross-reactivity to the delta antigen. He thought there was something else there. And you can see that the people who looked at people with acute hepatitis and chronic persistent hepatitis, and as you can see the people with more persistent hepatitis, you can see the delta antigen got positive. Then he developed an immunofluorescence assay based on that, and you can look at the liver cells and find out where the delta antigen is. And this was if you stain for core antigen, immunofluorescence. So it's actually in the nucleus, and they were lined up. So a lot of people did not believe that this existed at the time. So I think we make a point of reading a lot of journals, up-to-date journals, but I think people should go back and read old works or so. So this was a work done by Robert Purcell, who has discovered many hepatitis viruses, including this one. He got interested in this one, and then he had a chimpanzee colony. So those days we used to use chimpanzees as animal model to infect them with hepatitis B. So he had several chimpanzees who were infected with hepatitis B, had chronic hepatitis B, had resolved hepatitis B, all sort of things. So we took the sera from people with delta positive and delta negative and infected these chimpanzees to try and see and prove the Cox postulate. So this is quite complicated, but you can just see in the bottom line here are the delta antigen positive. So this is a chimpanzee. There are several chimpanzees in this. The one I put over here is the one already had hepatitis B surface antigen positive. And then you can see these dark bars are the ones staining for delta antigen in the liver. So so you can see the delta antigen start coming up here. You see the delta antigen viral RNA goes up, and then once that comes down, the antibody to delta goes up as well. So he really proved that you can actually infect chimpanzees with this particular virus that gave origin to the hepatitis delta virus. So inoculation of serum of delta positive donors infected chimpanzees. Delta infection did not result in chimpanzees with pre-existing surface antibody. So you could protect them if you already had a resolution of hepatitis B. You need chronic hepatitis B to be happening. Chimpanzees with pre-existing surface antigen, like the one I showed here, develop rapid delta infection and suppression of HBV replication. So delta appears to be a marker of transmissible pathogenic agent, either as a HBV variant or another agent that requires helper function for hepatitis B is defective in interference with HBV infection. So that's something that proved the emergence of delta infection. Now we know a lot more about delta. The delta antigen actually gets into the hepatocyte by using the NTCP receptor because its surface is exactly like hepatitis B. Once it gets in there, then it becomes the nuclear capsid goes in, uses polymerase enzyme of the host to replicate, and it makes two different types of delta antigens, the small and the large one. The large one, the small one is active replication, and the large one is involved in packaging. There is a step that's important for the large antigen, which is called prenylation. Prenylation is a post-translational modification of adding a farnesyl group to that large protein. It is very critical for the function of the large protein. If you cannot get farnesylated, the large protein will not work, and the packaging of the delta virus will not happen. So that's one of the potential targets for treatment. We'll come to that in a second. So then once it is assembled, it is packaged within the hepatitis B genome with the surface antigen sticking around and released into the bloodstream. The geographic distribution of HTV is quite complicated. Most people actually don't test for HTV. There are two reasons you don't test. First, people, you do initial testing, and because if you just look at general population, you don't find any, and you think, oh, we don't have delta. The second part is that people say, oh, what is the point of testing? Because I don't have anything to offer. So why tell people you have another terrible disease I have no, unless you have a problem with advanced liver disease. So either way, there are some places you have. So Northern Africa, some parts of South America, of course, Central Asia, such as Mongolia, and also Turkey and Middle Eastern area are all known pockets of hepatitis delta infection. Who you screen makes a big difference. General population is actually very low, but if you look at people who with cirrhosis, advanced disease, and have chronic hepatitis B, the yield actually goes much better. And this is also a targeted population. If you are from a particular place, then they do have, in Asia, I think Karachi has a very high incidence of delta population as well. So low middle income countries and low income countries do have a lot of hepatitis delta. The transmission is through contaminated needles and unsterile medical procedures. Prevalence is about 1 to 2%. And the prevalence is higher for people with advanced liver disease who undergo hemodialysis, similar to hepatitis C. The childhood vaccination can prevent delta, and there are studies from Italy where there are high incidence of delta. Childhood prevention actually brought it back down. So that's a good prevention. If you prevent hepatitis B, you can do that. So there are two ways you get delta: superinfection and co-infection. Most people know. Co-infection is when you get both B and delta together. When you get co-infection, you actually have both of them resolve over time, and that's usually the case. Very rarely they become chronically infected. But if a superinfection is the most common for chronic delta infection, you have chronic Hep B, and then you get another exposure to delta, and invariably most of those patients will develop chronic delta on top of hepatitis B. So screening for people, anyone with hepatitis B, is what common strategies use, but this is really not very well done. There is not even, United States don't even have an FDA approved HTV RNA testing, which is a concern. CHP patients are at risk of HTV, people from some high endemic country. Universal screening is very low. It's also not very cost effective to screen everybody if the yield is very low. So the logic is that if you have universal screening will work if you have a low HPV prevalence and have a high HTV co-infection prevalence. If you have the other way around, then you're going to spend a lot of money to find the very few cases. Diagnostic algorithm in acute and chronic stages is similar. You test for hepatitis B, and the hepatitis B is positive, you test for HTV RNA, and if the HTV RNA is positive, then you have delta as well. Otherwise, you just have chronic hepatitis B. Same thing with the chronic hepatitis B, you rule out other causes that would cause chronic hepatitis, and then hepatitis B is confirmed, you can look for RNA, and if your RNA is positive, RNA is negative, and the delta antibodies are positive, then means you have resolved delta infection. So unexplained hepatitis and unexplained progression of liver cirrhosis are the one you usually look for. Acute hepatitis exacerbation of liver disease is where that usually presentation of delta is. It interferes with hepatitis B replication. Most of the dual infections interfere with hepatitis B infection. So if you have Hep C and Hep B, the Hep B viral load is lower. Same thing with delta. The hepatitis B viral load will be much lower when delta dominates infection. So it is the most aggressive form of hepatitis that you can find, viral hepatitis that you can find here. Very aggressive process. 30 to 70% will have cirrhosis at diagnosis. And if you don't have cirrhosis within 5 years, they develop cirrhosis. More than 50% have a liver-related mortality in 10 years. So it's a very aggressive form of, if you have it, it is very difficult to treat, and we don't have limited treatment options. So you can see the same thing here, the normal liver, about 75% within 15 years go on to develop chronic cirrhosis and complications associated with that. So we want to look at what's driving force of hepatitis delta from an immunological perspective. So Arshid did the study by looking at two different groups of people, people with co-infection and people with mono-infection with hepatitis B. We had to go and establish this cohort in Ulaanbaatar in Mongolia. Another one was in Istanbul in Turkey. So where we had these patients collected over time. So chronic patients have hepatitis, they assigned, most of them match up very well in terms of ALT elevations and sex ratios as well. What she did bulk RNA sequencing followed by phenotypic characterization, and then did a re-stimulation protocol developed using our own peptides for hepatitis delta and hepatitis B, looking at HBV, HDV specific responses. Also looking at cytotoxic functions against hepatitis delta and proliferation capacity. Plasma markers also really looked at cytokine beta assays, looking at what are the cytokines you can find out that will contribute. Not going to go into the details of this, but she was able to look at generalized expression of the genes and the patterns, and the pathways are more pro-inflammatory and fibrogenic and carcinogenic pathways that are highlighted in the dual infection group compared to the mono-infection group. Inflammatory cytokines like TNF-alpha, the TS17 cytokine, IL-17, CXL2, CXL13, and TGF-beta are all elevated if you have dual infection, also contributing to the factor that these are cytokines that can lead to liver fibrosis as well. Studying this is complicated. Our institute really does a lot of work in characterizing humanized mouse models. So we generate our humanized mice model where we can actually take an NSG background mouse who has no background, and then you reconstitute the immune system using CD34 positive hematopoietic stem cells. So now you have the mouse which will have human lymphocytes. You can also do developmentally, you can destroy the liver of the mouse and actually put the implant in the fetal liver cells, and now you can have a human liver. We have done it with multiple different organs, but this particular thing, once you have that, now you can actually use that to infect hepatitis B and study a lot of things. It's not a perfect model, but it allows us to study the real aspects of hepatitis B as well as hepatitis delta, and you can establish that in these kind of models. So what she did, she'd actually be able to do that. Days after inoculation, the humanized mouse model does have HBV DNA go up, and as well as HDV RNA. And here also you can detect HBV surface antigen and D antigen. So this is an ongoing work that we will be able to now establish and study out progressive time. The mouse model is not perfect. It does not develop HCC that easily. So that may be there are some caveats to this particular model, but you can understand a lot more about liver disease progression. The work has shown multiple different pathways that involve cell cycle, p53 metabolism, Hedgehog signaling, and DNA damage, all contributing much higher in the co-infection group as the ways that HDV will induce and lead to carcinogenesis in these patients. Treatment, preventive action, of course, if you prevent hepatitis B, you're also preventing hepatitis delta. The treatment used to be all interferon alpha with very limited success, and most studies have shown very dismal response rate, and there is no particular cure. People have to be on interferon alpha for quite a long time. So coming back to the life cycle, there are several therapeutics that are now available, and that's why the renewed interest in hepatitis delta. You have NRI inhibitors such as Mercledex B, now marketed as bulevirtide. It's one of them, is already approved for use in Europe, awaiting approval in the United States. It blocks entry of hepatitis B as well as hepatitis delta because they have the same entry mechanism into the hepatocyte cells. You actually have interferon that works at various stages. You have RNA interference agents and monoclonal antibodies also being developed. And you have the prenylation inhibitor developed by Jeff Glenn that blocks the prenylation or farnesylation of the large HD antigen that will also affect the replication. Nucleic acid polymers from a company called Replicor, which is a little bit obscure action, but it seems to affect the packaging of the virus, also has activity against both HBV and HCV. So the endpoints are difficult for hepatitis delta. The endpoint, complete virological response, is that lack of detection of below the level of quantitation of HDV RNA is very difficult. We don't even have an assay that is quantifiable for the use in clinical trials with the FDA. We're developing that at this time. So that's one main concern. Partial virological 2-log decline, that is fairly accepted. Biochemical normal ALT levels, but the most people actually do is the combined response for clinical trials. If you have a 2-log drop and you have normalization of ALT, that is considered a good thing. Again, remember that compared to the other viral hepatitis, this is not a cure. This does not give you a cure. This actually gives probably a rush the rapid progression of the fibrosis. That's all. Going to give some time on. None of these is actually going to lead to cure. So let's go with one by one. Bulevirtide, NRI inhibitor, the PS1 analog, competes for hepatitis surface antigen to the NTCP receptor and blocks the entry of both B as well as delta. So if you give virological response to bulevirtide, 2 different doses here, you can actually 2.5 into 2-log drop of HDV RNA over 48 hours. Biochemical people, about half of the people who have normalized ALT by that time in 1 year, and with the combined response of 2-log drop and normalization of SD seen about half of the patient. Bulevirtide can also be combined with pegylated interferon, and that can also give you more sustained response. That is been used widely in Europe, particularly in Germany. The second target is the Rep23109. It's a Rep nucleic acid polymer. It has activity against hepatitis B as well as delta. And here you can see that delta, it has a good, all the marks points here are delta and RNA that is going down when you give replica 22319 in patients with hepatitis B. So it also can bring you down, have a good virological response in patients with the delta antigen small. But this is not something, there is no ongoing studies with 2319 now that towards an approval of this particular drug. But lonafarnib has. Jeff Glenn is one of the first agents that has been described with activity against delta as a prenylation inhibitor. Lonafarnib, if those who are interested, is already approved for the use of progeria, an orphan disease. It has the same mechanism which will reduce, progeria is accelerated aging. With the little kids with this disease, so they get aged very quickly. So prenylation inhibitors like lonafarnib is approved for that. But for this one, it has the same mechanism of action here, but it can prevent HDV RNA replication. The people who are screened, the treatment is for 24 weeks, and you follow up for another 24 weeks. You can see that about 3.6, 3.18 log drop is seen with lonafarnib. The problem with lonafarnib, it is not poorly bioabsorbable. So it has to be combined with ritonavir. Ritonavir is an anti-HIV protease inhibitor, but it's mainly used because it's a very good CYP3A inhibitor. So if you ever use Paxlovid for COVID, that's the same mechanism. You use that agent to combine both of them together. And then many times you also combine with pegylated interferon. So if you do that, you actually have a very good response rate, about 3.8 log drop with the 3 drugs when you combine with this medication. The problem is side effects. Lonafarnib has side effects, ritonavir has side effects, and a huge drug interaction profile. And pegylated interferon, as we know, has a lot of side effects. So the pathway for lonafarnib has stalled a lot because of many of these reasons, although it does have activity in replicating. So 3 different strategies have been developed to treat hepatitis delta infection. The strategies result in significant decline in HDV RNA levels. It's really not clear which of these will lead to functional cure. That means functional cure by definition you stop, and 24 weeks later you should not have any DNA, in this case RNA, and that's what the definition is. So we are not there yet, and none of these methodologies or any treatment has gone to do that at all in patients yet. So that's something to wait for and see whether there will be new therapeutics available. Some strategies require additional treatments like interferon, and that will increase the adverse event profile. Imagine many of these people are asymptomatic, and you give this really toxic chemotherapeutic regimen. It's really not going to be compliant on this treatment for a very long time. Bulevirtide is an injection, so you have to do an injection every day. So these are various challenges in uptake of this treatment in patients with that. One of the interesting things is that even though bulevirtide can block hepatitis B entry, it has no effect on surface antigen of hepatitis B. It's quite interesting that it's actually only at the entry level. It has no effect on cccDNA or any other HPV replication, which is quite disappointing because that drug was developed for hepatitis B, and then Stefan Urban, who developed the drug, repurposed it for Delta, and then sold over to Gilead Sciences. So the correlates of the clearance, we don't have any correlates of clearance. What if you're on suppression for a while? Who are the patients who actually can stop? It's really something that we really don't know. And if you can develop some parameters and an algorithm that if you follow this algorithm, you can stop, that'll be very useful in stopping HDV treatment. So Ashri's project was just to look at that. So she went and collected people who actually cleared viruses and then wanted to look at immunological correlates of spontaneous HDV clearance. And then hopefully that used a predictive algorithm that can be translated in the clinical setting with the therapeutics. So now you have a cohort of co-infected patients. You have a cohort of patients about people who are resolved hepatitis delta. And you also have a cohort of people who had resolved both B and delta. So you looked at PBMCs, looking at the phenotypic markers, RNA-seq, T-cell expansion, stimulation, restim assays I told you before. And she looked at finally, she also looked at single-cell RNA sequence to understand the logistics of this. So just to quickly go through, the HDV resolved patients are very highly activated central memory cells. So there is a very active immune system present, particularly in the CD8 compartment. That was clear, that much more significant in the patients who were clear that Delta virus than the people who actually had both hepatitis B and Delta. So antiviral, looking at HDV specific response is the big part of this project. For that, she took PBMCs and stimulated them with either hepatitis B overlapping peptides or hepatitis delta overlapping peptides. It's co-stimulated with CD28, CD49D, and then cultured them with IL-2 for 1, 4, and 7 days, and then at day 9 we do a re-stimulation, because the frequency of response is actually very low. Unless you do re-stimulation, you cannot bring that up in any kind of meaningful way. So you have to re-stimulate, and then you arrest with Golgi export by brephyridine A, and then you look at flow cytometry for detection of antiviral cytokines. So HDV-specific, the large amount of work, I'm going to show you the big highlights. HDV-specific CD8 positive T cells exhibit higher cytokine responses secreting interferon gamma, TNF-alpha, and the TFH cytokine IL-21. Immunologists generally believe, without any clinical data, that if you make more than one cytokine against a particular pathogen, that's really better than making one cytokine. And so here in this case, many of these people who have resolved infection really exhibit large number of cells that excrete interferon gamma and TNF, interferon gamma and IL-2, interferon gamma and IL-21, interferon gamma and perforin, which increases associated with the cytotoxic potential of the cells, granzyme B, again cytotoxic potential, TNF-alpha and perforin, TNF-alpha, granzyme. So very broad response and very elaborate response. It's primed towards cytotoxic and antiviral efficacy of this drug. Then she performs single-cell analysis, again looking at resolved infections and the chronic infection people. Combines very complicated analysis in terms of how many samples were involved and how many group samples were involved, where you can actually look at the cluster genes and what are the genes associated with that. Overall, the antiviral genes, in summary, large amount of work summarized that antiviral genes go up. The cells are activated and ready to go. T cell signaling pathways are much hyperregulated in these patients. Cell co-stimulatory molecules go up, antigen presentation go up, exhaustion signals do go down, and pro-fibrogenic genes go down when you have resolution of delta and in the co-infection rate. All these are exciting news that we could develop probably an algorithm based on that. If you look at the plasma cytokine levels, most of the cytokines that are elevated in dual infection, that are the pro-inflammatory cytokines and that lead to contribute to the fibrogenesis and carcinogenesis, are down-regulated when the delta infection is actually gone, and that allows you to have a much more quiescent immune system and arrest the progression of fibrosis. So many of these cytokines are elevated, or most of the cytokine tests are reduced in patients. So what is the profile, immune profiling of the subjects? Effector cells actually are significantly reduced in dual infection. They go up. The effector cells go up as part of a resolution of the delta infection and the delta and chronic Hep B infection. Exhaustion markers go down. Polyfunctionality increases, antiviral cytokines are produced much higher levels, activation patterns come up in a good way, and they also proliferate much better. All these markers provided really good algorithm we could develop clinically that would be, along with the clinical markers of RNA and ALT, you could develop something that patient can be stopped. So I'll end by saying that spontaneously resolved HDV patients exhibit qualitative and quantitatively improved CD4 and CD8 phenotype compared to chronically infected patients. The polyfunctional T, CD4, and CD8 cells responses and decreased anti-inflammatory cytokine profiles associated with HDV. A predictive algorithm associated with HDV clearance will be invaluable to guide HDV treatment. And at some point, the chronic HDV patients are going to receive the treatment. When can you stop this treatment? What can guide part of the duration of the treatment? So most of the work in the immunological work was done by Arshi, and that we collaborated with ND as well as Shihan, Yuvaruddin and Talid, and one of the people who in our group, a gastroenterology fellow, now is attending, Amir Abu Talib. He really championed these efforts of establishing these cohorts, and thank you. I'm happy to take questions.